So welcome everyone to April's Immerse Yourself. Thank you for everyone joining us here in the library and everyone joining at home over Zoom. I'm going to go ahead and talk a little bit about our upcoming events and speakers, and then I will turn the floor over to Ian. So first of all, I do want to thank this month's sponsor, the Dutton Law Group. So I believe he is in the room with us today, so thank you very much for your support. Upcoming events we have for May 4th and 5th. I know a lot of you are looking forward to Vintage Dive Weekend. We're going to have people going out in the water with vintage gear, just like the ones that Jacques Cousteau used, or if you're a Sea Hunt fan, the ones that they used with double hose. We're going to have live demonstrations with Mark V and Miller Dunn diving helmets in the water as those double hose dives are going on. So that's going to be on May 4th. May 5th, we're going to have more double hose regulators in the water on the reef. So those are two different dives. So be sure to mark your calendars for May 4th and 5th and sign up for Vintage Dive Weekend. On May 15th, so about a month from now, we're going to have our May Immerse Yourself with Dr. Joe Deturi. He is a U.S. Navy diver, saturation diver, and deep sea submergence designer. He's going to share his 28 years of experience in that field and kick off the opening of our new exhibit, Salvaging the Deep, which focuses on deep water salvage operations through time. And that's going to be May 15th. Tonight, our speaker is Ian Combs, who is standing right next to me. He is a senior biologist and coral nursery manager with Mote Marine Laboratory. He's going to talk a little bit about the work that Mote Marine does restoring coral in the Florida Keys, as well as the greater Caribbean, as well as the role of crabs in that conservation process. So without further ado, I'm going to turn the floor over to Ian. Ian?
Thank you.
Wet the wire. Wet the wire.
Oh, please. Well, first off, I want to thank you for the introduction, but I also want to thank all of you for coming to hear me chat a little bit about some of the amazing work that we do at Mo. I was just talking to one of the attendants in the back about our campus in Sarasota. And I think it's always really inspiring just to see the scope of all we're able to accomplish, not only here in the Keys, but also through a lot of West Florida. As mentioned, my name is Ian. I'm the senior biologist at Mote Marine Lab, and I'm the coral nursery manager down at the Summerland campus at IC 203. So my specialty is really in the care, the propagation, and the health of the coral that we grow on land, managing the obviously all the day-to-day operations of our nursery. And then my formal training is actually in this technique called structure from motion photogrammetry. But I'm not going to talk about any of that today. I'm going to talk about the coral. I'm not going to talk about photogrammetry. I'm actually going to talk to you about kind of a novel approach that Mote's taking in restoration. That's looking at holistic restoration, and I'll get into much more detail about what that means. I don't think I need to really stress on the importance of coral reefs, especially to people who live so close to them. However, I like to dote on my favorite ecosystem a little bit. This is an image taken in the local reefs. You got some grunts and some wrasses there. And this is a really good kind of depiction of just what's going on in a coral reef. And the short notes is it's a lot. Coral reefs are some of the most productive ecosystems in the entire world. People talk about coral reefs as the rainforests of the ocean. And the more apt description is actually that rainforests are the coral reefs of land. They're incredibly productive. They're incredibly efficient. Despite making up a really small percentage of our actual ocean landscape, they harbor somewhere between 20 to 30% of all species found in the oceans, and a quarter of all biodiversity found in the ocean. So this really small kind of biome contributes massively to global health. If you were to take all living coral reefs and put them into one area across the globe, it would take up an area about the size of Colorado. And that's, so to put that into perspective of just the scope or lack thereof of what these ecosystems inhabit and the amount of production that they're able to produce, it kind of blows your mind. And again, these are hyper productive, and if I can't convince you to care about coral reefs, maybe I can say, do we care about breathing? And like, so coral, like open ocean ecosystems are responsible for most of the oxygen that we breathe. I know growing up, we'd always see like trees produce oxygen, and that's true. But overwhelmingly so, our open ocean ecosystems produce the majority of the oxygen we breathe. And if I can't convince you to care about coral reefs, maybe I can convince you about breathing. And so we don't end up in this situation, if you guys remember the Mel Brooks film Spaceballs, where air is this commodity, then we should definitely care about coral reefs. And coral reefs kind of act as that canary in the coal mine to the general health of the rest of our oceans. So they might not produce the majority of the oxygen. The open oceans do that. It's much more expansive. But more locally, they do produce some pretty important functions to not only our day-to-day lives, but to ecosystems. Whether that's recreation, diving, street diving museum, whether that's supporting fisheries or just tourism. And if you can't appreciate the economic importance of coral reefs, maybe we can appreciate some of the more physical functions of them. Coral reefs actually do a heck of a lot to save our homes that are on these islands from wave energy, especially in hurricanes. Coral reefs can absorb, disperse, and relocate up to 90% of wave energy that they receive. So imagine as hurricanes as we get hurricanes in, if we didn't have those reefs, we know, especially the Hurricane Ian and Irma, how we can see that wave energy. Imagine how worse it would be if we didn't have a reef essentially blocking for us out on the ocean. And I mentioned hurricanes, and that's actually a unique strategy that coral reefs actually adopt. And it's this term in ecology called a disturbance-mediated ecosystem. So you can see this is clearly not a coral reef. However, these are not unique types of ecosystems, but a disturbance-mediated ecosystem actually requires these periodic disturbances to function and to thrive. If we didn't have these periodic disturbances, in the forest case, it's fire. In our case, it would be a hurricane. You would actually not see the amount of biodiversity that you do see on the reefs now. What would happen is competition would take over. And I hesitate to use the word survival of the fittest, but you kind of see a homogenization of the life, because those that are better at competing would gradually take over. Periodic disturbance comes in and kind of levels the playing field and allows some of these, say that again?
It's a trust buster.
Yeah, it kind of allows these other organisms to at least take a hold on and to take advantage of some of the space that's exhibited. Well, then, if these are disturbance-mediated ecosystems in a world full of disturbances, why would we expect them to not be thriving like that, right? Why do we expect them to look like this if disturbances are their game plan, but in fact they look like this? And that's due to a couple of factors. The main one is probably the fact that these disturbances are becoming more and more frequent and more numerous, right? So not only is it the hurricane, it's the global warming, it's the ocean acidification. I mentioned that they're really productive ecosystems. They're also really efficient, which means that for every gram of carbon that enters this ecosystem, 0.8 grams of that are used up and used within the system. That only means there's about 20% of the net intake that's left to be used for other purposes, or that can be lost without having consequences. That some of that net carbon leaving might be overfishing. We also have to deal with ocean acidification. So increased CO2 in our atmosphere actually leads to higher or lower, more acidic conditions, a lower pH in our ocean waters, which is a problem not only for coral reefs, but for a number of organisms. This past summer's thermal stress event is another example of a stressor where we're seeing increased sea surface temperatures, which is causing coral bleaching. And then we have other additions to that, which are point source. Well, this is a photo from somewhere in Miami, which is effluent being broadcasted into the oceans. Unfortunately, I think these are pretty few and far between scenarios as far as these point source ones, but these are things that we have dealing with global scale stressors all the way to extremely local scale stressors. And these disturbance mediated ecosystems go from something like this, which can handle a disturbance every 20 years or so, to looking like this, which is now we're seeing some of these opportunistic creatures come in. So we've gone from this very productive, very coral dominated ecosystem to something that's mostly dominated by sponges, soft corals, and algae. And I also want to make a note that these are very important on a healthy reef as well. But as we're seeing some of this coral decline, we're seeing some of these take over. And this in ecology is known as a phase shift, or a shift from alternative stable states, which is in class a great word to turn into an acronym. And this kind of really simplistic model is really what's going on with our reefs in the Keys. And obviously, there's much more complexity involved, but this really breaks it down well to bare bones of what we're seeing. So on the y-axis, we have coral cover, and on the x-axis, we have grazing intensity. So I mentioned that we have these algal-dominated reefs. Before, when it was a normal ecosystem, we have a variety of grazers, many of which you could probably see here, that are eating a lot of this algae. As ecosystems begin to shift, we begin to see a shift not only in the benthic cover, but in some of the stuff that's causing these, and you get this cascading effect, which we call a phase shift. So again, I say we have coral cover on the y-axis, grazing intensity on the x-axis, and these black bars and these white lines aren't there just for a nice graphical representation. These actually represent these thresholds. So once you cross one of these, for lack of a better term, the ball ends up rolling down the hill on its own. It no longer needs these different types of pushes to get there. And this is kind of the sweet spot that we were in the 1970s. So we had incredibly high coral cover and pretty high end grazing. That's where we'd all love to be right now. And if anyone was here in the 70s, you probably met people that were here in the 50s and said, no, those were the healthy reefs. And that's actually true. That's what's called a shifting baseline, where our memory of the sphere that we inhabit becomes the normal. And then that's a really dangerous game to get in as we get through time, because then degradation becomes the gold standard, and it's really difficult to go back and be able to know where were we 10, 20, 30, 40 years ago. To 1970, we had really high grazing pressure and really high coral cover. It's a great place to be. But toward the 80s, we started pushing back from that grazing intensity. And that was mainly due to the die-off of the long-spined sea urchin or Diadema antillaria, which is, people like to refer to that as a keystone species. A lot of the work that we're going to talk about today is actually the work of my supervisor and of the coral restoration, or the restoration program manager, Dr. Jason Spadero. And he really likes to give those air quotes to the keystone species, and we'll talk about that more. That's mostly because it revolves around his favorite organism, which is the crab. But we'll get into that. And so 1984, we lost the long spine sea urchin. And that's really when we kind of stopped seeing the Keys be the Keys as we remember it. And as you can see, that arrow graphically represents crossing that threshold, but that was the point where that stable state began to shift from the coral dominated stable state to the algae dominated stable state. And that was about in the 90s. We also saw a large die-off of some of our reef builders, which were the saghorn and elkhorn, due to a white syndrome that swept through the Keys and the Caribbean. And this is kind of where we've middled for the last 20, 30, 40 years or so, is in this really low coral cover and really low grazing pressure. These slides are out of order. So how do we get that shift away from that? There's a couple different ways to do that, and probably the way that you're most familiar with, or the way that you might think of first when you see the Mote badge, is through active restoration. And that is the active growing the coral, putting it back out in the reef, and replacing that coral cover that we lost. And that's what we do really, really well here at Mote. This is my home base, which is down in Summerland Key, so Elizabeth Moore International Center for Coral Reef Research and Restoration, eloquently named. And this is North America's largest land-based coral restoration facility. So we also have 2 other land-based facilities in the Upper Keys. We have one at Bud Mary's, and we have one at Reef House in Key Largo, as well as 4 offshore nurseries in Key West, off of Sand Key, Blue Key, right off of where Summerland is, and then 2 that match the, we have one in Islamorada, and we have one in Key Largo. And this is where we do the bulk of our propagation and outplanting of the coral, that active coral restoration. We've been doing so in the Keys for about 20 years, really, really successful. The idea is we grow them either offshore, like you see in these trees, or on land, as this picture shows, on little pucks. We grow them. We all know the genetic individuals of them, and then we outplant them on the reef. In this case, we're looking at a staghorn, Cropra cervicornis. We put them in arrays. All these little clusters are the same genotype or genetic individual. As they grow, they'll grow into each other because they recognize each other, creating a larger colony than if we were just to put one single coral out at the beginning. So just to reiterate, all of these corals are the same genetic individual. And then over time, they will fuse into each other, creating an area much larger in much shorter amount of time than if we had just put a single coral out there. And we have success stories. This is actually a photo from Belize, out at Laughing Bird Key, where before, I think they've been outplanting at that site for almost a decade now. And you have these beautiful cropper fields, obviously staghorn surrounding this beautiful elkhorn colony. And more locally, we have a success story. This is a photo of the first restored or outplanted corals to fuse and to spawn. I talk about all of this work of getting coral as big as possible, as fast as possible. Does anyone know why? No? Okay.
So
Reproductive maturity.
Exactly, yes. So I had a tour come into IC2R3 the other day, actually yesterday, and they asked me a question. They pointed a coral in my tank and they said, "How old is that coral?" It's maybe this big around. And I said, "I have no idea." And that's because, for a couple different reasons. As we frag them, we're asexually reproducing them, right? So it isn't, we don't know when the coral was born in our tanks because it's essentially cloning them. But 2, we don't really care. I don't know if that's the right word. But the goal is we want to get them to reproductive maturity. And the way that coral gets to reproductive maturity is through size, not age. So you can have a coral that can be 400, 500 years old. And we used to have those in the Keys, and we have those elsewhere. And it could be this big, it could be 400 or 500 years old, and it'll never spawn. Because whatever the genetic mechanisms that happen, until it gets to a certain size, it'll never be reproductive. So we try and use and harness the coral's biology and physiology in our favor. So we know that if they the same genotype, they'll form into each other. We know that if they get to X amount of size, depends on the species, that they'll start spawning. And we actually had one of the first recording instances of this. These were outplanted in 2015, and they spawned in 2020. Now, that's 5 years of what would take a coral from a larval stage, from sexual reproduction, maybe 40, 50, 60 years, if ever, given our current conditions. So it was a huge success. I was actually in the water the day before this to see them spawn the first time. It was my 3rd week on the job, 4th week on the job. So it was not only really inspiring to kind of like get into gear as far as like what our mission is, but it's a really great success story to show that this, like, our what was kind of theory up until this point is actualized. It was really cool.
Is that for the stag and elkhorn as well, or just the?
So,
Stone.
we have spawning colonies. We, as far as the reproduction goes, which is not my expertise, we actually have spawning colonies that we grow on trees that we bring back in. So we know that they're of the right size. That way we can, mix the, we know the genotypes of the parents, and we can enter them into our sexual reproduction pipeline. But the idea is not to have to do that. The idea is to essentially put ourselves out of jobs. So to get the reef back to reproductive function, because right now it's reproductively extinct. And so to start doing that, so essentially have the coral do our jobs for us, where they spawn, they seed the reefs, those grow, etc. So our reproduction team works with something like 7 or 8 different species. A lot of them are the staghorns, the elkhorns, and then the massives like root brain coral, the great star coral, things like that. I apologize, I'm really bad with coral common because I'm trying to get better. But I would like to note, that if we're just using active restoration, right, that's a really long way to go. That's a lot of work to go from our coral to pauperate state, which is a fun word to pull out at Scrabble on the weeknights, to the grazing tissue. So I'm going to propose to you, what if we're able to actually increase the grazing pressure? All of a sudden, that threshold, as far as like active coral restoration goes, becomes a lot more attainable, a lot easier.
And crabs.
That's a little foreshadowing. We're gonna go with the less sexy stuff first, which is the manual removal. There are programs within the world. I believe this is from the Great Barrier Reef, where they're actually physically removing algae from the reef to make room for some coral. We can stop removing grazers from our reefs, or we can culture grazers. Again, this is the long-spined sea urchin, and this is one of our own, Martin Mote, who essentially worked for decades up in the Upper Keys to kind of crack this aquaculture code that was the long-spined sea urchin. And this was his life's work, and amazingly, he basically was able to figure it out and crack the code. And then he said, "I'm retiring. I've been doing this for a while." And he passed a lot of his research on to Josh Patterson. So that's who's standing kind of in the middle to the left, in the light colored shirt. So he's a researcher at UF, and he took a lot of the stuff that Martin pioneered here. And was able to further it, and now they can basically successfully culture urchins at scale. So that's always the big question that we're always racking our brains with. Whether it's coral restoration, whether it's mangrove restoration, terrestrial restorations, how do we do this at scale? That's always the really exciting domino to fall when you can figure out how to do this not once, but dozens, hundreds, thousands of times. And you can see here we have some juveniles on the bottom. We have some larger reproductive size colonies in the bottom left. And then I kind of earlier did the air quotes around keystone species, right? And this is where we get to talk about diversity and resilience. If we were to put all of our eggs in one basket, all of our money into culturing, say for example, diadema, long-spined sea urchin. And then something like, I don't know, something that might happen in the 80s happened where a bunch of them die out, where are we left, right? So now that we have a choice, and in fact that the fact that the diadema in the 80s were the dominant grazer probably was a pretty good indicator that those reefs weren't doing great, right? Because you want we always say the word that a diverse reef is a resilient reef. So the fact that the death of the long-spined sea urchin, the mass mortality of the long-spined sea urchin was kind of the thing that tipped that stable state over the edge was probably a good indicator that the reefs at that point, despite looking really healthy, and maybe to us or the shifting baselines, probably wasn't doing great. Enter crabs. So this is a beautiful individual, a face that only a mother could love. This is the Caribbean king crab. Just as a caveat, the king crab people don't like us to call it a Caribbean king crab. They're definitely not related, but they're up in Alaska on the west coast, so we don't have to tell them right now. It's also called the Channel king crab, West Indian spider crab. And we are going to get into some taxonomy right now, which I will say I'm not a great taxonomist, but bear with me, the reward is decent at the end. In the 1800s, this was called Mithrax spinosissimus. And then sometime around when Jason was writing his thesis, they decided to change it about midway through his thesis so he could do a control F and have to replace everything in his dissertation. And they changed the name to Damithrax spinosissimus. I'm told, despite being a good Catholic schoolboy, never took Latin, I'm told damith or dammit is actually a word in Latin. I took Spanish, doesn't mean anything in Spanish. And then a year later, it was renamed to Magwamithrax spinosissimus. Does anyone know where Magwamithrax? Katie might, she watched. Say that again?
It's bigger.
Close. It's not close. It was an homage, and this is in the paper, to Tobey Maguire from Spider-Man. So the surname of Tobey Maguire, they named it as an homage to him. Taxonomy is pretty boring. You gotta have a little bit of fun. So this is Jason with his king crab. Say that again?
You're easily entertained.
Yeah. All the genetics that go into renaming and all of that, at the end of it, you'll be lucky if you come out with sanity. So a Tobey Maguire joke at the culmination is probably getting away pretty good. And they looked at these grazers. These are some of the most prolific grazers on the reef. You might not notice them very often because they're pretty nocturnal. I've seen them a few times dive in, but it's mostly when I'm like looking into a crack, and you can see them kind of scurrying out of the light. And they did a couple surveys and found that the natural density of these crabs is about 0.001 crabs per square meter, and it's like a crab every kilometer or something, which is low. And basically, we asked ourselves, can we shift that? Can we move that decimal point over and increase the density of these crabs? And what would happen? They actually looked at that not too far from here. I believe the patch reefs they worked at were like right off of Vanos, the Cuban place. And they worked in these patch reefs. And these patch reefs are actually wonderful experimental setups because they're these kind of large things of structure or hard bottom separated by pretty, we call them lack of stepping stones. So these pretty wide alleys where things typically can't, migrate too far or leave their home ranges. So it essentially, I mean, you can see these are boats, essentially provides a mini replicates of very different treatments all within one pretty easily accessible area. This is kind of similar to what those bombies look like from the sat view. And this is basically how they found them when they got there. So this, you can see this is what's referred to as halometa. It's a calcareous algae. Look like little hexagons attached to each other. So they grow calcium carbonate, not a whole lot of things like that. And it's one of those things that as the coral decreases, comes in and kind of takes over. As you can see, this used to be a beautiful coral head, and it's covered in algae now. And they put about whatever it was, I think it was like a couple hundred crabs in that area, enough to reach that density that they were looking for that square meter. And they also scrubbed them, or Jason scrubbed them, or an intern scrubbed them. And when they had a bunch of treatments where things were either left alone as controls, just had crabs added, were scrubbed, or crabs were added and scrubbed. I think there was an animation there. And this is what they found as far as macroalgae. So the first lines of control didn't touch anything. Macroalgae was pretty high. Obviously, there's some variation depending on various things or grazers that might already exist. But when you add crabs, you see a pretty stark reduction. Let's see if I can move. On the y-axis, we have cover, so the percent of macroalgae covering that area. And the y-axis, we have time.
Oh, wow.
So middle line, crabs, they do a pretty good job. And then the bottom line is scrubbing plus crabs.
What's the time on the bottom?
I think it was a year.
Yeah, and it was, I think they did it a year every month.
Be sure to restate the question in case somebody else can't hear us, please.
Some people online.
Yes, we have a bunch of people online.
This is gonna be a kind of an obvious question, I think, but so are the crabs eating the algae?
Correct, yes, they are herbivores. We don't think of, sorry, the question was, are the crabs eating the algae? If you have anything you can throw at me, if I don't repeat the question, feel free.
Okay.
Yeah, so the question was, are the crabs eating the algae? Yeah, kind of unique amongst, as I don't know, as we think of crabs. I'm from the DC, Maryland area. I grew up with blue crabs, yeah. So, you know, detritivores, things that eat meat. However, a ton of the crabs that are exist in the Florida Keys are actually herbivores, and the king crab is no different. So they're actively grazing on the algae. The really nice thing about these crabs is that they don't have huge grazing preferences. So if it's green, I mean, they have their preferences, but if certain fish or whatever, if that type of algae doesn't exist on the reef, they'll go somewhere else. Crabs, not so much. If it's there, they don't have what they want, they'll eat it. If they get hungry enough, they'll eat it. So it makes this kind of like the, you know, being the obligate grazer that it is, makes it a pretty good situation if we're trying to do things like, you know, reduce the amount of algae on the reefs. So an 85, yes, go ahead.
Well, tell us about scrubbing then.
Yes, the question was, they asked about scrubbing. That's kind of exactly how it sounds. Like that photo that I showed of the vacuum. It's just a physical removal by a person, a human, a diver, of the algae. So whether it's physically scrubbing, depending on the type of algae, it can be that. But for the most part, you're down there, you have a giant bag, or 2 or 10, and you're getting as much algae as you can, throw it in a bag, shipping it up to the deck, and then most likely throwing it away when you get back to wherever you're trying to go. So yeah, and so the combination of the two. So obviously you would hope to see grazing or the algae cover decrease when you're scrubbing. But imagine, you know, in a normal coral reef, imagine having like a yard where you have a bunch of features in it, like rocks or trees or something where grass doesn't grow. Mowing, that's probably, minus getting around everything, probably not too hard. Now imagine trying to mow a football field where the algae or the grass grows back faster than you can get rid of it. And that's essentially what these reefs are as far as from a human perspective. You would have to dive all day, every day, which probably sounds pretty fun to a lot of us, but doing that forever is a lot of work. I don't know if, as a grad student, having a result this drastic and this like obvious, you go to your advisor and they basically go, cool, do it again. Like, I don't believe it. It looks too good to be true. Let's do it again. That's exactly what they did. Experiment up the keys a little bit, but same type of design. We had scrubbed, non-scrubbed crabs, and then scrubbed with crabs. And they did it at fewer time intervals. And that's mainly because Jason was sick of driving from Virginia to the Keys and back every month, which I would also agree with. And again, we can see very similar trends. So the top is the control, and then we have our various treatments, and it's mostly downward trends, which is incredibly rare in these types of ecological experiments because there's so many different variables that are affecting your data. So to see this happen with this drastic of a result multiple times is huge. Go ahead.
One of the lines showed an increase at the end.
Yes.
So what line is that, and do you know the cause of that increase?
That is the crab line. That could have been a number of different things.
I think that's the ones that were just scrubbed. and no
Just scrubbed?
crabs, Yeah.
So,
But people got tired of scrubbing.
They only scrubbed in the beginning of the experiment, I believe. And so first was scrubbing on July 2015, and then after that they didn't scrub every time. So that's just showing how much algae came back after.
Same location as well, or did you have
Uh,
to move to a different test site?
we went up the key, so a little further north.
Different
So that
test
line
site.
was just scrubbing and no.
And they just scrubbed it and then we
No
were
crabs.
monitoring. Mhm.
Oh, so you would expect it to come back, okay.
Not to, sorry, Kate, but you forgot to repeat the
Oh,
question.
sorry.
Thank you.
Sorry for.
And this is actually a kind of a photo from a before and after throughout this experiment. So while the algae looks about the same, I would reckon that maybe the fish or something that pops out to you. That's actually something that they also looked at. So they looked at fish abundance, both, through time between the different treatments. So on the y-axis, we have the number of fish in the frame from the survey. They were either taking video or taking photos. And then we have our different treatments on the x-axis. So again, we have control, scrub, crabs, and the combination of the two. And this first one is looking at before they applied each of these treatments. So essentially the baseline. You can see they're about the same. I don't think there's many significant differences between these two statistically. So you can kind of look at these as all about being, the same amount of fish abundance at each of these sites. And then you add the treatments to them, and you can again see drastically different things between these and the control. And most notably that the control is about the same pre and post. And just one note about the scrubbed, I asked Jason about this, and that much higher in scrub is actually due to like a site effect. I think the scrub site was much bigger. And a lot of these fish have really large home ranges. So that's that. But the takeaway is that removing algae increases fish. And these are the 5 most abundant fish groups that were in there. And the really interesting thing about this is about 3 of those are actually grazers themselves. So not only is getting rid of this algae promoting fish to come back to these reefs, it's promoting grazers to come back to these reefs, which you would think would then have even more synergistic effects with grazing. And then the richness of fish. So richness is just the different types of fish that come. And that saw an increase with the treatments as well. So now we're getting into a lot of this, what we call like return to ecosystem function with these crabs. So not only are they doing something that we want as coral restoration practitioners to basically make room for us to put coral out there, but they're also bringing other things in to make some of these jobs easier. If we can get more grazers in than what we originally put out, it even saves us from having to scrub even more. And finally, they looked at juvenile coral density. So this was not a restoration experiment. So there was no outplanting of the coral put on there, but then they did surveys and looked at how many coral were recruiting to these sites. And this is corals per meter squared on the y-axis. And again, we have our treatments in here as well. And as you can see, the density of juvenile corals is increasing as algae decreases, which is huge. Yes.
So these are natural occurrences of new coral growth, not outplanting.
Correct. Yes. So the question was, are these natural recruitment of corals, or are we putting coral out there? And no, this is before Jason's time at Mote. So this was a purely ecological study. No outplanting occurred at some of these studies. However, this is informing our outplanting and coral restoration strategy moving forward, which we'll get into.
There's a place for them to land. There's a place for them to.
Exactly. So as far as algae being a bad thing on a coral reef for a coral restoration practitioner, the things that we're really concerned with algae are twofold. One, lack of space, right? So if we put coral out there, we can do that. But if you have a coral larva that's swimming in the open ocean, trying to look for a place to settle, it can't find a good hard substrate, a good piece of rock or hard bottom to latch onto. And then even if it does, which is a pretty in the reefs here, especially those coral or those algae dominated reefs I showed you, that's really hard for a coral to do. Happenstance that it would happen, then what happens is the coral actually smothers it and covers it and blocks out the sunlight that the coral needs for its photosynthetic algae to help it grow. And a third, probably lesser known contributor is a lot of these algae. Some of the reasons that they aren't eaten up is that they produce these really gnarly, like secondary metabolites, like basically chemical defense to keep from being eaten, which is working. And so depending, I think I'm not great on the literature of that, but that's probably one of the reasons they're not getting eaten. And if there's disturbances, I wouldn't be surprised if those are also affected. But that's definitely why they aren't getting eaten as much. Like that dictyota, that two-fork stuff, can produce some pretty gnarly things. And the halimeda, which was the calcareous one, fish just don't like to eat chalky, rocky things. So most of them. Yeah, so actually parrotfish are a lot pickier than you would think. We're doing ourselves a lot fair, which is why we love the crabs. So these crabs, we've talked about this Caribbean king crab is this, obligate grazer. It's really not picky. It'll eat whatever it's put out for it. So if we can use this synergistically with our coral restoration to bring that algae cover down, also increasing our fish abundance, increasing our fish richness, and increasing our coral recruitment, we've all of a sudden got a really interesting platform to add to our active restoration that we've already been doing in the Keys. And again, this is a nice before and after picture of one of those patch reefs, where you can see this is a Orbicella annularis head. I apologize for the lack of common name knowledge, but all of that is some of that calcareous halimeda.
Mountain star, maybe?
That's Flagellata because it's
Flagellata, yeah.
No, I go through this a lot too. And this is after, so you can see all of a sudden not only have we opened up a lot of space, but all of the algae is effectively gone. Hopefully, we can return to this beautiful, thriving, vibrant reef that we first displayed at the beginning. So we mentioned this before. Incredibly strong potential for coral reef restoration, which I think is a really important distinction, right? So coral restoration is the act of putting coral onto a reef. Coral reef restoration is the act of returning ecosystem function to these reefs. And it brings in much more than coral. We talked about it brings in the crabs, grazers to get rid of that, brings in fish, etc. So a much more holistic view of our coral reefs rather than either physically removing algae, which we've talked about is probably pretty laborious, and helping us along to create a better environment for when we are able to go put coral out on these reefs. So what we need now is more crabs. We talked about the density of those crabs naturally being about 1 every kilometer or so. I believe for these, they put 500 crabs on these reefs. And I think Jason had to go collect 500 crabs for these treatments, which doesn't seem like fun. But we've acknowledged that this is a transformative and successful mitigation strategy for this algae. And who else acknowledged it is Mission Iconic. I don't know if you guys are familiar with Mission Iconic Reefs, but it's an initiative by NOAA and the National Marine Sanctuary to pick, I believe it was like 7 iconic reefs throughout the Keys and return them to something like 25% coral cover in 20 years. And the reason that 25 is significant is in the 70s and 80s, that's about what our coral cover was sitting at. Nowadays, it's sitting at about 1%, or even after the thermal stress event, probably a little lower than that. And these are the 7 iconic reefs. And I think we've estimated that we would need about 28,000 crabs per reef to enable, or per unit to restore these. Go for it.
So the crabs are staying in the area, they're not migrating out?
Yeah, so I believe these slides were left out because we've still got a ways in this talk and I'm running out of time. They did two different things, but they looked at the crab home ranges and they stay pretty close to those areas. They know that the food's there, they like it. But the other key is we'll get into the aquaculture side of things. They found as a survival metric, they basically have to get the crabs to be about 30 millimeters wide. And their predation decreases. Yeah, so you don't have to grow them that big. Like all the pictures we've had of the crabs are these big, giant crabs and holding them. You only have to get them to be about this big before their predators decrease.
Can you repeat the question?
Yeah, sorry, appreciate it. Could you remind me of the question?
The crab stay.
The home range of the crabs. The question was, do the crabs hang out at these reefs? And the answer in so few words for me is yes.
Yes.
And they also do not get predated upon until they reach a certain size. Appreciate that. This work was also done in Belize, in the Mesoamerican Reef, which is the second largest barrier reef, the third largest barrier reef being the Florida's coral reef, and the first being the Great Barrier Reef. And the estimates are we need about 96 million crabs in order to restore and basically restore these reefs and to see that treatment on a reef-wide scale. So how do we do that? We talk about how scaling up is always an issue with us, and essentially the name of that game is sex or breeding these crabs. We talked about how Martin Moe spent decades trying to crack the code of the long spine sea urchin. Relatively, these king crabs are an aquaculturist's dream. They only have to exhibit 4 or 5 different life stages before turning into a juvenile, many of which don't even have mouth. The spiny lobster that we, as a huge fishery, I think that goes through something like 26 larval stages. They have to go out to the open ocean, come back. So as far as complexity in growing these things, it's pretty straightforward, pretty easy. This is one of our wet lab and one of our hatcheries at IC2R3. And this is where we became really successful in kind of the piloting of how do we culture these, how do we get them going. The other nice thing is they kind of eat whatever. We throw any sort of algae we find. We're restoring the reef, when we're opportunistically go on a reef to do some scouting or whatever. We see a bunch of algae, we bring it back, we give it to the grazers lab, and they love it, they grow their own. And we have a bunch of little crab babies that are hanging out. And so we become pretty successful at not only ensuring that they become reproductively active, but that once they reproduce and once they broadcast their larvae into the ocean or into the water column, that we can rear those successfully. And that's a little guy. Yeah. So that little lab space that I just showed you, there's no way that we could deal with the millions of crabs we need in order to restore. So we actually have a larger hatchery at our marine aquaculture park, which is kind of it's 20 miles inland from our City Island campus. And we repurposed some sturgeon aquaculture labs to grow king crabs at scale. These are giant sturgeon tanks that were used for basically trying to aquaculture sturgeon for caviar. And we've dug a couple up. We've kind of made sure that they are ideal for crabs, not sturgeon. And this is how we're going to get to those millions of crabs. And we know that as the math works out, we can successfully get to about 28 million crabs in about 10 years from this facility. These tanks are so big that in order to go count the crabs, you have to go diving in the tanks. So these are two aquaculture staff that are up there. And this is another known tank that's gravid. They're releasing their juveniles into the water column. So I'm a little surprised that none of you called me out so far. At the beginning, we talked about monoculture, keystone species are putting our eggs in one basket. So why am I just talking about Caribbean king crab? One, we know that it's easy to culture, but 2, we're also recognizing that that's not the sole answer to this question. And we're actively looking at a number of research options, and a number of things of bringing different species of this Mithracidae, which is the higher order that the Caribbean king crab belongs to, and using those. And a bunch of studies have shown that these actually are pretty voracious herbivores as well. And they're all found in the Keys and in our own backyards. So these two species, which I wrote down the genus names, I kept forgetting it as I was practicing this talk, and I wanted to get it right. So on the top, it's Mithraculus carife, and below it is Mithraculus cinctomatus. There's a quiz at the end of this.
Well done.
Spelling test at the end of this. Yeah.
Oh, no.
Yes.
Traditionally, these were found around staghorn colonies. Now that we don't have a ton of staghorn, they've kind of migrated in these seagrass fields and varieties fields. But we're looking at introducing these into our restoration along with our Caribbean king crabs, and using those as a synergistic outplant with our staghorn. And these might look similar size to the Caribbean king crabs, but in reality, they're about this big. And we know that like pound for pound, they actually outgraze some of the bigger stuff. I know they're this big because I've been sent on like a half a dozen dives where they're like, hey, go find these for me. I can't. They're too small, they're too fast. We're also looking at kind of trying to optimize the herbivore assemblage. So not only using the Caribbean king crab, but using these other species in tandem with our long spine sea urchin. And one grazer that we're really leaving out is the snail, and looking at seeing if we can co-culture these with some of our recruits too. So using snails, and we can culture snails, and they eat a different type of algae. If we can do that. These are actually recruit size corals, so like really tiny guys, and looking at different assemblages of snails and how they graze in and around those coral recruits on these tiles. And lastly, maybe penultimately, does anyone recognize the spiny lobster's cousin to the left?
Spanish, a lot of Spanish.
Yeah. So while the spiny lobster gets all the accolades with the fishery here, these guys are actually some of the most voracious predators on the reef. And it's basically a twofold thing where studies have shown that these lobsters we know these lobsters communicate via urine. However, we know that some of these grazers actively flee when they can sense these spiny lobsters' urine. So looking at different entities, we know that's true with some of these urchins. We're looking to see if that's true with some of these crab species. And then if it is or not, I think some of the preliminary studies have shown that, you know, when the presence of the spiny or the spotted lobster urine, these guys stop grazing and start fleeing. So it's counterproductive to what we want. But that also leaves a really interesting management question, because as far as these voracious predators go, they might be one of the only things that eat some of the corallivorous snails that eat our outplants on the reef. So it's a really interesting, like, how do we sort this out? And so I hope through all of that, we at least got an appreciation of kind of taking from that very simple diagram of the grazing pressure, coral cover, and just how actually complex these systems are. And how we're actively trying to work to tease out some of these intricacies, but also use all of this to have a more synergistic restoration effort to complement our already, tried and true methods of our active coral restoration. So restoration is a team sport, and we really can't do this without you guys. And so as far as Mote goes, we have a number of ways to get involved. Katie, correct me if I'm off base with any of this, because she's the expert. And I think made these part of the slides. Yeah, nice, because they're way better looking than Jason's. So if you want to get out and dive with restoration, you can go to our website or at Scuba Pro's website and dive with Scuba Pro. We also work really closely with iCare, which is a local based initiative that actively participates in active coral restoration with many of our coral stocks and we work really closely with Mote or with Scuba Pro, excuse me, and that's actually them diving one of our offshore nurseries. You can volunteer with us, both in the Upper Keys, and you can come hang out with me if you want down in Summerland. I'm pretty fun to work with, I promise. Despite what some of my employees might think, but I'm pretty good.
Is that in a dry environment?
So the question was if that's in a dry environment, and yeah, so as of right now, we're working to integrate more diving programs into our normal volunteer workflows. But as of right now, dry. You're going to be hands in tanks. You're going to be actively, scrubbing the coral and working with the coral, fragmenting the coral. So we try and get, as far as volunteers go, like anything we can do, we try and get you guys trained up to do that as well.
So a non-diver could do something.
A non-diver could absolutely do this. Yes. And we also give tours of our facilities, both in the Upper Keys and in the Lower Keys. So if you want to go to mote.org/keys tours. I know that the IC2R3 tours are Tuesdays at 10. Katie, can you remind me what the Key Largo and Islamorada tours are?
In Alvarado, Bud and Mary, we do them on Tuesdays at 2, and then in Key Largo at Reef House, Monday, Wednesday, and Friday.
So
Also
if you
schedule
want to get
private
4
tours.
out of 5 days, or schedule a private
Yeah.
tour on Thursday, you could have a Mote tour every day of the week.
There you go.
Oh, and minus the private tours, all of these tours are free as well. We just ask that you sign up online so we can get a head count. And we also provide a couple different hands-on experiences. Do you want to talk about this?
Sure. So Slice of Paradise is a paid program. On the other side. Slice of Paradise is a paid program that our education department runs. And so you can get hands-on experience microfragmenting your own coral. So this was $125 per person. But it's a great experience, and all that money goes directly back into our coral restoration programs down here.
And for those people that are online, could you announce your name and how to contact you?
Oh, yes, I am Katie. I'm the community outreach coordinator at Mote in the Upper Keys, and you can find all this information at mote.org/keys tours or mote.org/volunteer, or you can email me kcleek@mote.org.
Kay is also my saving grace in this portion of the talk.
I think that should be it.
You can also buy a Protect Our Reefs license plate. So this is actually fun timing, like POR license plate. I think it's a $25 donation, and something like
Generates a little over $1 million a year in income for us, and we also grant out about a quarter of that to other scientists and organizations that are starting to do coral restoration stuff as well.
So the reason I'm forgetting all of that is because I've spent the last week preparing a presentation of funded research for this to present tomorrow at the POR symposium. So if I have bags under my eyes, that's because I've been crunching data and then figuring that out for the last week to present research that was actually funded by these license plates. And you can donate. If you want to scan the QR code, I'll wait. But lastly, I hope that not only with Katie saving me on a bunch of these questions, but none of this happens without in a vacuum. Not only me standing up here talking about my supervisor's research and our program director's research, but having volunteers, having tons of people. We also have tons of partners that are outside of Mote, whether that's the National Marine Sanctuary, Fish and Wildlife, state, federal partners. We have tons of university partners that we work with. So none of this happens in a vacuum or on our own, whether it's with you guys volunteering with us or with our collaborators outside. And I believe with that, take any questions. That's not my name, but I go back. Any questions? Yes.
I have a question on the algae that you gather.
Okay.
Is there any value to that for fertilizer or any?
Yeah, so the question is there any value to the algae that we remove from the reef, other than feeding the crabs that we're growing on land? So that's a really tough one. I mentioned all those weird secondary metabolites. You go to the beaches and the sargassum washes up and creates a hazard. They really don't know what to do with it. It's a huge portion of active research. I know that there's work. Oh, I forget who the group is. I think that's out in like the Costa Rican area where they're trying to create fertilizer from it. They're trying to do a bunch of different things.
T-shirts.
Insurance. I don't know if anything's like landed as far as that, but it's something that we're all trying to figure out. Or also crab food for later. So if you find some, I'm gonna drop it. Yeah. Other questions? Yes, in the back.
I found it interesting that you got more recruitment of herbivores on the reefs that had the least amount of algae. You'd think you'd get more herbivores where there's more algae.
Sure. The question was, they were curious about how there was more recruitment of fish to these areas where there was less algae, and you think that the kind of counterintuitively, you think that if there was more algae on the reefs, you'd have more herbivores. And then, just to be clear, we're talking about like herbivorous fish, correct?
Sure, absolutely,
Yeah. So my best guess at that, and I think it's a fairly educated one, is that the algae that was removed is probably some of the nastier stuff, and it allows for maybe some of this early stage successional algae or some nicer algae to colonize when the gnarly stuff was removed by the crabs to promote some of this fish. The other possibility would be that the reason that it stayed low is because those grazers were grazing that and keeping it low, kind of iteratively. And then potentially a third one would be that by some of those large like overhead shots of before and after, in the after shots you could tell there was a lot more like depth and a lot more structure to the reefs. Some of those really small herbivores really crave that structure. When you get algae kind of taking up all these niches, you get essentially a flattening out of some of these reefs. And so it could have been a physical structure change. By removing some of this, you're kind of promoting more hidey holes for some of the fish to come in that, like, you know, especially those small like wrasses and don't love being in these open ocean environments, or even above, they stay down low. So potentially that could have been a reason as well.
One more, I guess, could be, they go where everybody else is. You see other fish there, it must be some reason to go there.
Yeah, and so the repeating that, it was that maybe it was just they knew that more things were there. And that's really interesting because there's been a bunch of work done about bioacoustics on reefs, where like people have played sounds of healthy reefs in not healthy reefs, and more things recruit to the sound of a healthy reef, whether it's healthy or not. And obviously more things recruit to healthy reefs. So that could be a very good, like very plausible explanation too, is maybe this healthy reef, just like everyone was there, it sounded like it, and so they all came. It's party. Any other questions?
I have two from Zoom.
Okay.
One is, are there any unintended consequences from the introduction of crabs? Are they and the urchins native to the reefs that you find?
Yeah, so as far as we know, so the question was on Zoom, so I don't think I have to repeat it. But in case, it was, are there any unintended consequences of introducing these crabs, and are the spiny sea urchins and the crabs native to the Florida Keys? As far as we know, we have not seen any unintended consequences yet, and probably that is because both of those species are native to the Keys. So it's not like a situation with the cane toad in Australia, where it's an introduction. Like these you know have been here for a really long time, and as at least with the spiny sea urchin, I mean both still exist, but we're kind of reintroducing them at different densities.
The other question from Zoom I had is genetic diversity tracked in the crab populations? So
the question is genetic diversity tracked in the crab populations? And it is. So right now we are not actively putting crabs out on the reef as part of the Mote program, because we have to go through a bunch of different health assessments and different basically certification courses. One of those is getting a better handle on some of the population genetics of our crabs, how different they are from different areas. All of the crabs that we come in and get, we tag with a unique genetic ID. It's a lot easier as far as genetics go to track individuals because crabs are not clonal like coral is. So like it's very rare that you have two individuals of the same genotype, or that are the same genotype, because that's a clone. However, understanding how mixed the populations are, because that's really important for things like inbreeding. So once we get a better handle on that, we'll work with the regulatory agencies to say, like, if we find out that they're very distinct populations, I'm sure there's going to be special, measures taken that we might not want to plant lower keys crabs in the upper keys, but we're still learning all of that. And I think there was one more question. Yeah.
Yeah, I mean, that was basically going on the crabs. I know you mentioned the spiny sea urchins and the Caribbean king crabs. But you also mentioned that there were a few other types of crabs under the same family of the Caribbean king crab. Are those also native down here?
Yes. So those
Okay.
two Mithraculus species are native.
Cool.
And I'm really bad at catching them. Yeah. Any other questions? No? Well, you can find me in the parking lot and shake me down if you have any more pressing. Thank you for your time, everybody. I really appreciate it.
So thank you for being here,
Of course.
and the History of Diving Museum, we have a ton of people. I know that last time I counted, there was over 30 online. So we have a bunch of people that weren't here tonight, but we thank you for your time, and we gift you a 1-year membership to the History of Diving Museum.
Thank you.
So when you're not working, you can come 362 days a year.
I'm not on a moat tour.
You can come to the
Thank
History
you.
of
I really
Diving
appreciate
Museum. So
that.
thank you
Yes,
very much. We
absolutely.
appreciate
Thank you
it.
so much.
And thank you all. I look forward to seeing you at Vintage Dive Day. We also have a Mark V dive raffle. The Dive into Art is closing tonight. And next month at our Immerse Yourself, we will be opening Salvaging the Deep. So thank you all.
Thank you. Thank you.